VHF.
Good morning, sir.
Hi, good evening.
Good evening, sir.
Yeah, good evening. See, can you able to see the screen?
Yes, sir.
Yeah. All have joined.
Two more people have joined, sir.
Okay, okay,
One more person has joined.
okay. Sangal, Tarun, and Nidish Ashkia.
Yes, sir.
Okay.
Yes.
Good evening, sir.
Yeah, good evening.
Hey, Nitish, what's the chapter for tomorrow's history test?
Age of Gupta.
Gupta, sir.
Yeah.
Okay. That Mauryan Empire, right?
No, it's Age of Gupta. That's it.
The Mauryan and age of Guptas are same.
I don't care.
Okay, shall we start then?
Wait, are they same?
Nah, they're not the same chapter. That's all I know.
Hello, students. Can you hear me?
Yes, sir.
Yes, sir.
Yeah. So, shall we start?
Yes, sir.
Yes, sir. Everyone
Yeah.
joins. Sir.
Okay, fine. Good. So, what is this chapter?
A new chapter.
Economic
importance of
of
bacteria and fungi.
and fungi. Okay, economic importance of bacteria and fungi. So first we discuss about bacteria, then we can go for fungi. Okay, so
Yes, sir.
I think with your cooperation today we can finish with bacteria. I hope so. Okay, we'll see. Fine. And in next class about fungi. Fine. Yeah. Before starting, you tell me what do you know about bacteria?
Sir, for me, your voice is breaking a little.
No, it's
Breaking
clear
up.
for us. Sir, for me it's clear, sir.
Oh, then for me, only okay,
Sir,
I'll just leave and join
bacteria
you.
are unicellular organisms. They are
Mm,
eukaryotes and they
mm.
are autotrophic.
Okay. Unicellular, single-celled organism, and it is not eukaryotes, prokaryote.
Yes, sir, that's what I said.
Prokaryote. Yeah, prokaryote. Without a true nucleus. Fine. And why it is called primitive organism?
So because it's in the first kingdom.
So
No,
now I
no.
can
Yeah,
hear
they
you.
evolved first. See, they evolved first. Okay.
So what did you ask, sir?
Why they are called primitive organism?
Sir, because they have not a well-defined nucleus like a prokaryote or
Primitive in the sense they only evolved first. Evolution of living
like our
organism.
ancestors.
Yeah, ancestor.
They,
Okay.
yes,
Evolution. They only evolved first.
so I, for
Okay.
me, sometimes your screen is not there, and then
Now,
That's
again
only
comes.
for you. For me and all it's clear, sir.
Okay, yes, sir.
okay,
Yes, sir.
fine, good. Next. And most of the bacteria, some bacteria are phototrophic, and many bacteria are not phototrophic because they do not have chlorophyll.
Yes,
Okay. And what is also not there? Not differentiated into different parts.
yes. Oh,
Okay.
like
Fine. The basic general thing. No special parts.
yes. 1 second,
Just
sir.
a single cell itself organism. Okay. Fine.
Yes, sir. But
Now,
it has like flagella and all. It's parts, right?
yeah. Such kind of components are there, which gives them some special property. Okay,
Okay, sir.
fine. They have single chromosome. Another important thing, they have only single chromosome. Okay, only one chromosome is there. Okay, and no nuclear membrane. So basic, these are all the basic thing you should know about bacteria first. Okay, fine. Next, where bacteria are present. If I ask what are bacteria, you can tell me unicellular, prokaryotic, single-celled organelle with no membrane-bound nucleus. No nucleus is present. Okay, and now
Yes, sir.
where bacteria are present?
So it's there in our body, it's there in the food, it's there in the sky, it's there in the floor, it's everywhere.
Yeah, everywhere.
Yes,
Bacteria is
sir.
present everywhere. In air, water, in soil, in foodstuff, on
Even in
or
our body.
inside the body. Okay, and everywhere, like God, everywhere they are present. Okay, fine.
Yes, sir.
Where bacteria, sometimes they may ask, in our body, where bacteria are present? In our
So
mouth,
it's present in
in our
the
skin,
secretions made by
intestine.
the stomach.
Yeah,
So
look
intestine
at
and
it.
stomach.
Yes.
So also in our mouth also it's there, right,
In
sir?
intestine, see, E. coli. E. coli is something called the common cells. Common cells means they live in our intestine. Okay,
Yes, sir.
fine.
The
So
bacteria everywhere.
in our mouth, in our skin, inside the intestine, genital tracts, in our body. Okay, they can be living. Okay,
Yes, sir.
sometimes the
So
intestinal
genital
bacteria,
tracts is mouth, right, sir?
genital, see urinary tract.
Oh,
In male and female, bacteria can give. Sometimes they may cause urinary tract infection also. Okay, we
yes,
get
sir.
fever, body chillness, etc. Male and female reproductive tract or urinary tract, the bacteria can live. Okay, fine.
Yes, sir.
Sometimes they may be beneficial or sometimes they may be causing also. Beneficial in the sense what? They synthesize vitamins such as vitamin B. Sometimes they may be causing diseases also. Okay, like diarrhea, skin infection, etc. Okay, fine. Now, the diversity of bacteria. What we are going to study? The diversity of bacteria. What are the few things which we discussed as of now? What are bacteria? Where they are present in our body, where bacteria are present,
Liver.
and then
Oh, like
what beneficial effect it has, and what pathogenic means, what kind of diseases it causes. Okay,
yes, sir.
am I audible?
Yes, sir. You're
Yes,
audible,
sir.
sir. Can
Fine.
you? Am I audible?
Yeah.
Yeah. Okay.
Now we are going to discuss about the diversity of bacteria. What we have discussed, all these things are commonality, general thing about bacteria. Now we are
Good,
going to discuss
bad.
about diversity. What is called diversity?
There are many types, variations.
So
Yeah,
like how
the
many?
differences. Yeah, fine. Now the size of bacteria, it range from actually 1 to 5 micrometer. Okay,
Yes, sir.
Yes,
what
sir.
is
We
the size?
studied that
1
in cell
micrometer to 5
chapter.
micro. Yeah, 1 to 5 micrometer. But the average size is what? 2
2
1
micrometer.
micrometers.
2 mic.
Okay, fine. 2 micrometer
2 millimeter. I
now.
will be able to see it, sir.
What is the thickness?
Micrometer.
0. Yeah, micrometer. 0.5 micrometer thick. Okay,
Yes.
long and thick. Okay, fine. So that's
Yes,
about
sir.
the size of the bacteria. And then the shape. What are the different shapes? 4 shapes. If it is round, spherical, it is cocci. If it is rod, it is bacilli. If it is spiral,
A curve.
spiralum. And
Sir,
comma
are we
shaped.
done with the previous
shape.
chapter?
Huh?
Are we done with animal kingdom?
Yeah.
Yes.
Yeah.
Yeah,
But
we
am
finished.
I audible?
Yeah.
Sir, but
Okay,
did
we
we?
have done.
Sir, but did we do like you know
Mammalia, stuff like that.
See, listen, yes, we have done with the animal chapter.
sir, did we do the last page, the class
Yeah, we did
Mammalia?
class
We
Yeah,
Mammalia,
we
did
did
right?
this.
We did class
everything we
Mammalia.
discussed.
Am I
Okay.
audible?
Yeah, you're audible, Pa.
Oh, okay, sir.
Yes, I can continue.
Pa, please read it. Once the chapters are finished, do your assignments carefully and do it. Okay, please read at home. Okay, fine. Now,
Yes.
so round shaped, rod shaped, spiral or twisted, and big or comma shaped bacteria. Okay, so round shaped bacteria are called cocci. Rod shaped bacteria, bacilli, spirilla, vibrio. Okay, so they are the
Vibrio.
common 4 shapes. And
Yes.
now, say, look at that. Please listen one thing. Every bacteria, for example, they can live as single. Single means what? Monococcus. Monococcus means what? Bacteria live as a single. Then diplococci. What is called a diplococci? Always bacteria lives in pair. Lives in pair. That is called diplococci. Or in colonial form. Okay, colonial means what? Bunch,
Group.
clusters.
Group, sir.
Look at that. Like clusters. You can see colony, group of bacteria, or chains, filamentous bacteria.
Yes, sir.
Yes, sir.
Monococcus, diplococcus, colonial form, filamentous form. So the bacteria, can you see the chain, filament bacteria? Diplococci, only 2 bacteria are present. Diplococci. Okay, colonial form. You can see this one and this one are colonial form. Okay,
Yes, sir.
and coming to bacilli. Bacillus or rod-shaped bacteria. Again, the bacilli also you can see monobacillus, dibacillus. You can see, and then filamentous bacillus,
Lactobacillus, like that.
colonial. So all bacteria can live in same. They can continue all this form: bunches, chains, single or double. Bunches, chain, single or double. Okay, fine.
Yes.
And then the spiral, curve-shaped bacteria. Okay,
Yes, sir.
and then you have comma-shaped bacteria. Comma-shaped
Vibrio.
bacteria. Yeah, Vibrio cholerae. Okay, fine.
Yes, sir.
So they are the shapes of bacteria. And now, structure of bacteria. Okay, what are the ultra structure of bacteria? What are the components are present? Okay, it is unicellular, and then bacteria. The outer one, which is called what? You can see.
So
You have
the cell.
look into the
Cell
structure.
wall. The capsule.
I just enlarge the picture. Can you see now?
So the image, sir, 9.2.
Yes, sir. Yes, sir. Yes,
Look at
sir.
that. So you can see the dark one. This is called what?
Capsule.
Well,
Capsule.
Just
Capsule.
in the cell chapter also.
See, the outermost protective layer is called what?
Capsule.
Capsule. And then cell wall, the yellow portion. The black portion is called what? The black portion.
Capsule.
Black capsule. And you can see yellow cell wall. Yellow, dirty fellow. Yellow cell wall.
sir, is
Yes, sir. Based on this image, I'm telling the colors are not universal. It is just for your verification. I'm telling capsule, which looks black in color in this picture, cell wall, which looks yellow in color, this picture, and then after capsule, you have protoplasm. Protoplasm
Sir,
consists
the
of
brown
what?
color one is protoplasm. Oh, sorry.
No. Yeah,
Oh, sorry,
all
sorry.
the components. The brown color one, brown.
The cytoplasm, cell membrane, and chromosomes.
Yeah, brown cell membrane. Inside to that cytoplasm. Sure, this is cytoplasm, and then
Yes, sir.
the DNA, bacterial chromosome,
Yes.
single bacterial chromosome.
So I had a very hard time
Okay.
learning this alone, sir.
Yeah, single bacterial chromosome. Okay, and all these things are component of what? Protoplasm.
Protoplasm.
Okay, capsule, cell wall, and then protoplasm. And then internally, inside the bacteria, what you should see is plasmid. What are called plasmids? Again, single circular DNA. Plasmids are single double stranded circular, like this. Single double stranded circular DNA. They are what actually called what? Plasmids.
As
And then many, like all the black dots are actually called what? Granules. Maybe glycogen, maybe fat, maybe lipid, etc. Okay. And
Yes.
then some bacteria will have flagellum. Okay, flagella. Okay, and then it may be single flagella, double flagella, or many flagella. Okay, different type of bacteria are there based upon the presence of flagella. Can you understand the structure of bacteria? Okay,
yes, sir.
Sir.
and now, so living substance called protoplasm lying within the cell membrane, like cytoplasm. It consists of protoplasm, consists of bacteria, cytoplasm, and bacterial chromosome, and then surrounded by a non-living stuff called what? Cell wall.
Wall
Okay,
made of cellulose.
yeah, and then cell wall is made up of what? Peptidoglycan, not of cellulose. Listen, bacterial cell wall is made up of peptidoglycan, not of cellulose. This is very important. Not of cellulose. Glycan.
Oh,
Okay. And then a thin cell membrane is present between the cytoplasm and the cell wall. Between cytoplasm and cell wall, what is present? cell membrane is
membrane.
this.
Sir, is this peptidoglycan made out of glucose or something like that?
Peptidoglycans made up of sugars, amino acids, sugar
Sugar is
amino acid
glucose.
combination. They call it as what? NAM, NAG, N-acetyl muramic acid, N-acetyl glucosamine. In the higher class, you study about this. Okay, I think
Yes,
we
sir.
studied in cell. Did we study
No,
NAM,
sir.
NAG?
No,
So
sir.
NAM,
No,
NAG
sir. No, sir.
substances. That's what. So peptidoglycan made up of different chemicals. Teichoic acid, and then disulfide linkages. It's a complex structure. Okay,
Very complex.
fine. So very polymer, many substances mixed together in the cell wall. Okay,
Yes, sir.
and no well-defined nucleus, but single DNA is present in the center region. Okay,
Yes, sir.
and
It's a
then
prokaryotic cell.
yes,
Yeah.
prokaryote.
All
Bacteria
bacteria.
or prokaryotes. We saw it already. Bacteria, Munira, we discussed.
Both both fungi. I mean, kingdom
Yeah,
bacteria and fungi
Munira,
are
we discussed.
prokaryotes.
Protista, we discussed. Fungi, we discussed. Plant, we discussed. Animals, we discussed. So
Yes, sir.
please go through the previous classes and
Sir, that
not
we are doing
enclose.
pi kingdom classification in our school actually. So
Okay.
it's
They
like
also
a
said
second time.
prokaryote only.
It's like a second time revision, sir.
See all these things. Please go through. We will revise it also. Fine. Okay. Fine. So somewhat attached to the cell membrane. DNA. The region. Look at that. Can you see? The central DNA is attached to the cell membrane. That's what is mentioned here. At some point. Okay.
Yes, sir.
And then what are the other substances inside the cytoplasm? You can see there are little vacuoles, granules, and some bacteria are having green pigment called chlorophyll. Some bacteria, very few cases, they have chlorophyll. Okay, if they have chlorophyll, what property they get?
So photosynthesis.
They become photosynthetic bacteria. And sometimes, not in all bacteria,
Sir,
but in
so
some bacteria, capsule are also present. After the cell wall, capsule is present. Not in all bacteria, but in some bacteria outside the cell wall. Yes,
Yes,
sir,
sir.
so you said that a few bacteria in few cases contains green color pigment, which means they can conduct photosynthesis, and that time if they can conduct photosynthesis, they can like make their own food. Like, but few
yes.
bacteria will not have pigment, and what will they do, sir?
Or autotroph means heterotroph. We are going to discuss about the nutrition of bacteria. Don't worry.
So they can
Okay,
be
chlorophyll.
like anything. They can either be autotroph or heterotroph.
Yeah. They can. There are different modes. We are going to discuss. Don't worry. There are different modes.
Yes, sir.
After movement, we will be discussing about this. Okay, don't worry. We will come. We are coming there. Okay, fine. Now, so bacteria general structure we discussed. Now the movement. See, only if they have a whip-like structure called flagella. Most bacteria cannot move. Keep that in mind. Most bacteria cannot move. Okay, maybe passively transported by wind, water, contact, etc. But some bacteria, if they have, so look at that, this is called flagella. So they can move. Okay, they can move only if they have flagella. So that is called what? In a liquid medium, bacteria can swim. That process is called what? Locomotion. Movement of bacteria.
Plural of flagella is flagellum.
Movement of bacteria through flagella is called locomotion.
No, sir. I said plural of flagella is flagellum, right? Plural, sir.
Plural. Plural, yeah.
Yes, sir.
Flagellum is singular. Flagellum is singular. Flagella is plural.
Oh,
Okay.
okay. Yes,
Fine.
sir.
Now only the questions which you asked. Nutrition. How bacteria eats. Most bacteria have no chlorophyll, so they depend on ready-made food. So they are called what? If they require food from different sources, they are called heterotrophic. And how they are classifying the heterotrophic organism? Saprophytic and parasitic. See, look at that. Saprophytic means they get nourishment from dead and decaying organism. Saprophytes means what? Dead and decaying matter eating organism. Okay.
Yes, sir.
Now, parasitic means what? Living host. Example, in plants they can live, in animals they can live, in insects they can live, whatever. So they are called what? Living host, parasitic organism. Draw
Yes.
nourishment from living host.
Yes, sir.
Yes, sir.
Okay, fine. So two type of bacteria. Then bacteria secretes powerful enzymes. How the heterotrophic organisms can live? Bacteria secretes enzymes. That enzymes digest food substances and then they absorb. Okay,
Yes, sir.
this is the bacteria. This is the bacteria. Okay, and then this is the food. Look at that. Food is bigger than bacteria. Okay, this is food.
Sir, where, sir? Are you drawing anything?
Yeah, he's drawing that. You can't see that
Yeah,
ball.
bacteria.
No, I can't.
You can't see that ball, that thing.
No.
It's written food also there.
Bacteria. Listen first. Don't worry.
Where?
No
In
big
the
error.
middle of the screen or in the top of the screen?
No, in top of the screen.
No, sir. All I can see is NAM dash.
Just listen. See, no big
Okay, Leon.
diagram there. Just listen first. Bacteria is very small. Food is very big. How bacteria can eat?
Break it down.
Yes,
Yeah,
sir.
it secretes enzymes. It produces a chemical. The chemical from bacteria reaches the food and breaks down the food, converts the food into simple substances, and then they absorb. Okay?
Yes, sir.
Yeah, that is the main thing. Okay? About nutrition in bacteria. Now, respiration. How bacteria manages the respiration process. Look at that.
Aerobic and anaerobic.
Yeah, if they require
Arabica.
oxygen, they are
Aerobic.
called
I
what?
said aerobic.
If they depend on atmospheric oxygen, they are called what? Aerobic. Okay? And then if they depend on, if they do not require oxygen, no oxygen,
Anaerobic.
they need no free oxygen, means oxygen free environment. They are called what? Anaerobic.
Anaerobic.
Anaerobic. Okay?
Yes, sir.
See, listen, if bacteria, aerobic bacteria in absence of air die, anaerobic bacteria in presence of air, it dies. Okay? That's about
Yes.
respiration in bacteria. Now, next process, reproduction in bacteria. Main method of reproduction is asexual method. Bacteria is a unicellular organism reproduced by what method? Asexual method. Okay?
Yes, sir.
And name of the method is called what? Fission or cell division. Name of the method is called what? Fission or cell division. Look at the steps. Please look at the steps. Can you see the diagram?
Yes, sir.
Yeah, this is a bacteria. First, what occurs? What difference in the second picture? What difference you can see?
Sir, it's longer.
Yeah, elongation. Can you see? Enlarged. Elongation occurred. And then, can you see? DNA duplication, elongation and replication. DNA duplication. Can you see here? DNA duplication. Initially, there was what color bacteria?
Blue.
DNA.
And black.
What was in color?
Black.
Black in color, correct? In
Yes.
the back here,
Yes,
it is
sir.
black in color. And then the duplicated copy, DNA duplicated copy is called what? In what color it is present?
Red.
Red color.
Red. Very good. Red. So means bacteria duplicated. What is the name of the process? DNA duplication process.
Sir, what is this? Cell division.
Is called what? DNA
Coding.
duplication is called what?
Replicate back.
Replication. Okay, replication means what? One DNA duplicates its another copy. Okay,
Yes, sir.
elongation, DNA duplication. Okay, elongation and followed by DNA duplication. And then look at that. Segregation. They call it as what? Segregation. See, you can see here one furrow forms. Can you see a furrow in between?
How can it copy DNA, sir?
That is the process called a replication. One DNA by enzymes. Enzymes perform that function.
That's what
Okay. One
starfish have
DNA
to do, right?
divides into two DNA. For the cell division to occur, the bacteria should replicate its copy. Okay.
Okay,
So now,
sir. So the one DNA will divide into two DNAs. I
Hmm.
thought the one DNA will create another DNA.
same thing only.
Oh,
Fine. Next, something called what? Segregation of DNA. What is segregation? Look at that.
Like they divide.
One bacteria this side means one nucleus, this side one means one DNA this side. Original DNA from the center it moved this side. Okay, and another duplicated DNA moved this side. That is the next one. And then you can see cytokinesis. What it is called? What?
Separation.
Yeah, cytokinesis means what? Cell division.
Yes.
Cytoplasm division. Okay, can you see the cytoplasm is divided? Look at that. Here,
Yes, sir.
cytoplasm of the cell divided and two bacteria. From one bacteria, how many bacteria you have? Two bacteria.
Sir, two.
So fission means what? Division. Fission means what? Division. Can you understand
Yes.
sir.
the process, the steps? Yes. Fine. Next step. The steps are explained here. In fission, what occurs? The circular DNA duplicates. The cell expands. Expands is called elongation. Circular DNA duplicates.
Segregation.
Cell elongates. And then two DNA pulled apart.
Segregation.
Segregation. That is called what? Segregation. Two DNA pulled apart. And then middle separation. Cell constricts at the middle. And then
Cytokinesis.
cytokinesis. This is cytokinesis. And then two daughter cells are formed.
Oh.
Okay. And then two daughter cells are formed. Okay. Can you understand?
Yes, sir.
Yeah. And so this is called cell division. But this is the major method by which bacteria divides. Okay. That's asexual mode of reproduction. Okay, that is called what?
Asexual mode of
Asexual
reproduction.
mode of reproduction. Okay. Now, sexual mode of reproduction. Okay, how bacteria is unicellular or multicellular?
Unicellular.
Cellular.
Unicellular. Can single cells have sex?
No, I think.
Actually, yes. There are also some kind of sexual reproduction occurs in few bacteria. In very few bacteria, sexual reproduction occurs. Okay. For example, there is a method called what? Conjugation. What
Conjugation.
is the name of the method? Conjugation.
Conjugation.
See, let me draw a diagram for conjugation. See, look at that. Can you see the one which I draw? This is one bacteria in the bottom. Can you see a bacteria? One round?
Yes, sir.
See or not? This is another bacteria. And now, here there is a DNA. This is what? DNA. Okay? What color it is?
Blue.
And now, one organelle called what? Sex pilus. Okay, one organelle called what? Sex pilus. The orange one is called what? Pilus. And only some bacteria will have this organelle. Okay, sex pilus. Okay. Now, this sex pilus develops into what? A tube-like structure. Tube. See, like this, a tube-like structure forms. Okay. What forms?
Yes,
Sorry.
sir. Tube-like
What forms, sir?
structure.
Tube-like structure forms. Okay. And that
Yes.
tube-like structure, with that tube-like structure, here the tube forms. Okay. The tube is called what? They call the tube as what?
Pilus.
Sex pilus develops into, see, listen, conjugation tubes. They are called what? Conjugation tubes. Okay. And you know one organelle which is present in bacteria. See,
Sir, nucleus, DNA,
see, listen.
flagella. Sir.
See, listen. Inside that, what the green color represents what? What organelle?
Nucleus.
No. This is blue color is no nucleus, that is single DNA. Okay, no nucleus, that is single
Yes,
DNA.
sir.
Right. Now, the green color represents what?
Chromosome.
A green color. There I draw in round. What is this?
Oh, it's the protoplasm.
Plasmid. Plasmid. Do you remember? Plasmid.
Yes,
Okay,
sir.
green color is called what? Plasmid.
Plasmid.
Plasmid. So, with the help of the plasmid, this DNA, with the help of the plasmid, the DNA is transferred into this bacteria. Can you understand? With the help of the
Yes,
plasmid,
sir.
the plasmid, okay, carries that DNA into this. You understand? So, this one DNA to another, means one bacteria to other bacteria, the DNA is transferred. So, this process is called what? Sexual mode of reproduction. Can you understand? That is what?
Yes, sir.
Yes,
Sexual
sir.
mode of reproduction. And here, who is the donor?
Sir, the
The male
sir,
bacteria.
it is sir the bacteria which is creating a tube-like structure.
Yeah. Here, this is the donor,
Pilus.
and he is the recipient. The first one is the donor, second one is the recipient. Okay, the hollow tube-like structure extension is called what? Conjugation tube. Hollow tube-like structure is called what? Conjugation tube. Can you understand how sexual mode of reproduction occurs? One simple method.
What is that positive and negative, sir?
Donor, the recipient. Positive one is the donor. Okay, donor is what? Positive one. The recipient is called what? The negative one. Just to differentiate.
Yes, sir.
Okay, the donor, the recipient. So that is one method of sexual reproduction in bacteria. Okay,
Yes, sir.
so what is the name of asexual reproduction method in bacteria?
Fission, sir.
Fission. Very good. What is the mode of sexual? One simple mode of sexual reproduction in bacteria.
Conjugation.
Conjugation. Very good, very good. Okay, and fine. The next method is spore formation. See,
Yes.
under unfavorable conditions. Listen, under unfavorable conditions. Okay, the bacteria dry up themselves. Okay, bacteria dry up themselves.
Yes, sir.
That time they draws its content into spherical mass. For example, this is the bacteria. Okay, look at the shape. This is the bacteria. But under unfavorable condition, like this. Can you see?
Yes, sir.
That part is called what? And they develop thick,
Yes, sir. The develop thick.
thick and hard protective wall. See, look at that. Here wall is thin, but here the wall is thick. Okay, this is called what? That rounded body, little rounded body is called what? Spore. Okay,
Will explode like and then
spore,
spread.
spore. That is called spore. That method is
Yes.
called what? Spore formation.
Spore
That
formation.
method is called what? Spore
Spore formation.
formation. So, what are the
Yes.
three methods we discussed?
Fission, conjugation, and spore formation.
Very good, very good. Okay, and then, but later, under favorable condition. Okay, under favorable condition, the cell wall ruptures. Under favorable condition only, the cell wall ruptures, and then they develop into new organisms.
Sir, how does
Is
this
it
even
clear?
come under
Can you
economic
understand?
importance, sir?
Yeah,
Yeah, you remember?
yeah, yeah, yeah.
Sir,
Yes,
it's
yes.
generally about bacteria only it's given, sir.
See, you must study about the general characters of bacteria. Then you can go for
Same
economic.
That we
slide.
did in last chapter, no, sir.
Yeah. So, but you should know all these things in detail, right?
It
Yes,
is
We
sir.
will rest,
economic.
no problem. See, did you study asexual mode of reproduction there?
Yes,
No.
sir.
You remember?
Yes.
Yes.
Okay. Okay. I will ask you another question now. Let me check. What are the other modes of asexual reproduction? Means sexual reproduction. Here we saw only one method,
Sir,
conjugation.
that's spore formation.
What are the two more methods? I think I would have told you two more methods of asexual sexual reproduction. What
Fragmentation.
are they?
So fragmentation.
No, that's
Sexual
asexual.
reproduction.
Yeah, asexual only, sir, asked me.
No.
Sir are sexual.
Two fragmentation in bacteria.
Oh.
Two more methods
So
of
after conjugation,
sexual reproduction. Do you remember?
one
No,
is
sir.
conjugation, two more. So
You don't remember? Okay.
Oh,
Sir,
sir.
did we?
Okay, they are called what? Transformation.
transformation.
And then,
I don't
and
even
then
remember seeing all of this, sir.
Sir, I think we
Transformation.
never did this, sir.
I did not
Okay.
teach it.
Sir.
Translocate.
So that's what. So then you should listen. Here only they are talking about sexual reproduction. So know about these methods also. Okay? Fine.
Yes, sir.
So just keep in mind, what are the other method of reproduction? Sexual reproduction?
Transformation and
Transformation and
transduction.
Yeah.
transduction.
I would have told, but now at least you'll remember transformation, friends, from dead bacteria to live bacteria. Molecules are transferred. Dead to live. That is
Yes.
another sexual mode. Transduction means one bacteria to other bacteria through viruses. Okay? One bacteria to other bacteria. Bacteria to another bacteria. Viruses transfers the gene. That is called what? Transduction. Okay?
What's transduction?
One bacteria to other bacteria, DNA is transferred. by virus. It is called what? Transduction.
So they need a virus to come back alive.
Virus will come here, take the DNA, then it goes here. And then to another bacteria, and then to other bacteria, another to other bacteria, and then to other bacteria.
So, but the
All
one
virus
the
come.
virus is taking the DNA from will die, right, sir?
No.
The
No?
bacteria will die.
Oh, alcohol.
Okay, it
It will
lyses
die,
the
yes, sir.
bacteria and come out. Okay, it
Yes, sir.
lyses the bacteria and come out. Okay,
Yes, sir.
sometimes see here, sometimes bacteria will win. They cut the viral DNA. Sometimes virus will win. They lyse the bacteria.
So, but
It
most of the
depends.
time virus will win, right?
Most of the time, bacteria kills.
Oh, kills the
Most
virus.
of the time, bacteria kills. Only sometimes virus wins.
So that means
If
this transduction
if
is not
always see,
really
listen, just
more.
imagine if always virus wins, no bacteria now. But more than viruses, bacteria are alive. Can you understand?
Oh,
Yes,
yes, sir.
sir.
Just form a
Fine.
group and kill all the
Okay.
virus.
Okay, fine. So now, what is the special aspect, the advantages of spore formation is mentioned here.
Antibiotics.
Tolerate dryness and
Cannot
viability
be
is
killed.
no, they cannot be killed. They can tolerate
Okay.
poisonous chemicals. Okay, just keep this in mind. Okay, and spores can be carried by wind, water, or contact, etc. On meeting favorable conditions, spores germinates. Okay, and then the protoplasm becomes active from dormant state to active, and then they form a new bacterium. They rupture the thick cell wall and forms a new bacterium. Can you understand? So that is spore formation. Simply escaping unfavorable condition. Okay.
Yes.
Now let's go to the useful commercial applications. The useful thing of bacteria. This and all. See, all the previous processes you need explanation. This actually you can study. Anyway, let me highlight. Okay, bacteria in different area they are used. First, medicine. In medicine, what are the applications of bacteria? Antibiotics, serum with
Serum.
growth factors, and vaccines. One by one, we will see. What are the three important contents produced from bacteria?
Antibiotics, serums,
Antibiotics,
and
serums, and
vaccines.
vaccines.
Antibiotics, serum, and vaccines. We will see these things. What is an antibiotic?
It's a bio.
Chemical
Okay.
substance produced by living organism can stop the growth of some disease producing bacteria and fungi.
Yes, sir.
Okay, chemical
Yes.
substance produced by living organism. That chemical substance can stop the growth of some other disease producing organism
Sir,
like bacteria and fungi.
are white blood cells antibiotics?
So they are antibodies. White blood cells produces, they are not antibiotics, they are called what? Antibodies.
Yes, sir. But
They-
I
Same
mean,
property only. See, same property only, but two different molecules. Antibodies different from antibiotics.
Yes, sir.
Yeah, same function only they have.
yes, sir.
Yes.
Now, antibiotics for bacterial viral infection. Example of antibiotics: streptomycin,
Cold.
chloromycetin, tetracycline, penicillin. Okay, what is
Yes.
about penicillin? Penicillin is the first produced antibiotic from a fungi
So
called
for what is it
Penicillium
produced?
notatum. What is the fungi called?
Penicillium
Penicillium
notatum.
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 Yeah, they only produce the chemical substance called what? Penicillin. This is discovered by Alexander Fleming.
So
Okay.
in a book, it's given as Penicillium chrysogenum.
Hey, that is different.
Yeah.
Same thing.
Again, see, listen. Another species, Penicillium chrysogenum, also can produce penicillin, but from Penicillium notatum only, Alexander Fleming
Oh, okay.
discovered penicillin.
So it's written so formerly known as Penicillium notatum.
Yeah, Penicillium notatum. that
Yes.
only produces penicillin. Initially, it was discovered.
Sir.
Okay, and then
Yes,
it can penicillin can suppress growth of many bacteria. Okay, and
so it's
then
made from a bacteria to
yeah,
provide. I mean,
it's called
depend to
the
bacteria.
fungus. Fungi. Penicillium notatum is a fungi. Okay. The next antibiotic is called what?
Streptomycin.
Streptomycin. Streptomycin, discovered by Selman Waxman. Selman Waxman. And what is the name of the organism? Streptomyces griseus. Streptomyces griseus. And now they have given examples of a table. Penicillin, source of organism, Penicillium chrysogenum. Look at that. Here, Penicillium chrysogenum, because now we use chrysogenum only because more effective than Penicillium notatum. And then for what disease they are used? Tetanus, diphtheria.
Yes, sir.
And then what is the Sangalp? You tell me, what is the second antibiotic? What is the source? And
Sir,
what disease?
the antibiotic is streptomycin. It was discovered by Selman Waxman, and the organism was the bacteria Streptomyces gerius.
Grises.
Jesus.
ISIS.
Treatment for tuberculosis. So like that. Next one, chlorotetracycline, streptomyces, arifacient, typhoid,
Patients.
and then erythromycin from Streptomyces erythraeus.
So we don't have to remember the source organism, right?
You have to remember. Okay, that's
Oh
why it
my
is
God.
given.
Since this is
Definitely
random, let
questions
us forming
are
a
asked
word.
from this. Okay, this table is must. Okay, must. You must remember.
Yes,
See,
sir.
the get of biology student is remembering the name of microorganism.
Yes, sir.
Okay, okay. You are a
Yes,
get
sir.
biologist only when you remember name of the microorganism, biological name of the organism. Okay,
Yes, sir. So
fine.
that means
Next,
penicillium
what
is
is
the genus
mm,
and chrysogenum is the species.
yeah? Okay, listen. So keep remembering read the table
Just
and
stop it and
practice the names, pronunciation as well as the spelling. Okay,
yes,
please
sir.
focus. Some 10 more minutes only. We'll finish. Next, what when you call antibiotic is a good means broad spectrum means one medicine, one antibiotic for many bacteria. Okay, one antibiotic for many bacteria, many pathogens. Then only it is a good. Okay, and then
Yes,
no side effects, no side effects, and then it should
no side
not
effects
kill
means
normal bacteria of the host. We have good bacteria. Antibiotics should not kill normal bacteria. Then if it fulfills all three criteria, then it is a good antibiotic. And then where we use antibiotic as food preservative, treating animal feed, controlling plant pathogens. Apart from human diseases, we treat
Sir, we can't
antibiotics
take
for.
antibiotic.
Yes, we are taking. Whenever you go to
no,
hospital,
you
The
should know that.
yeah,
turmeric is antibiotic, right?
yeah, everyone, even the newborn babies are given antibiotics. Okay? But they will check
Paracetamol is
whether it
not
is,
an
hey, listen,
antibiotic.
they will check whether body accepts or not. Okay, fine. Okay, now serum. What is the next one? Serum. What is serum? When you centrifuge the blood, see, listen, when you centrifuge the blood, what remains? See, the hot substances like fibrinogen and cells. Blood, serum means blood minus cells plus hot molecules. Okay? When you centrifuge the blood, see, listen, when you centrifuge the blood, the hot large substances settled in the bottom, and then here plasma is present. What will be present? Plasma is
Plasma.
present. Okay, from that plasma, if you remove one substance called fibrinogen, okay, when you remove one substance, it is called what? Fibrinogen. Okay, from blood plasma,
Yes,
you
sir.
remove also fibrinogen, minus fibrinogen, then it is called what? Serum.
Yes.
And what serum contains?
Antitoxin.
Antibodies and antitoxins. Serum contains antibodies and antitoxins mainly. Okay, fine.
Yes, sir.
Yes, sir.
What are toxins? Poisonous protein released by bacteria. Toxins are poisons released by
An
bacteria.
antitoxin or not
Okay.
poison?
Antibiotics are medicine for the toxins.
Oh,
Okay. Antibodies are medicines for the toxins. Okay, fine. So just keep that in mind. That's what is given in this. Okay, serum contains antitoxins, preventive measure. Okay, and then small dose preparing serum, like small dose of bacterial toxin, something called vaccines. How vaccines are produced? They take serum, from that they isolate a toxin. Serum, they take serum, they isolate bacterial toxin, they inject into horse or cow. Understand the step. First serum, and then bacterial toxin is isolated, then injected into horse or cow. And then in their body, after several injections, the blood is taken out. Okay, fourth step. After several injections, blood taken out. And then serum again, the serum is separated. From that, antivenin means produced. From that means the medicine are produced. Can you understand?
yes, sir.
That's the fifth step.
Yes, sir.
Okay,
Poor
fine.
horse.
That's how many some vaccines are also produced. And we can, in genetic engineering means, instead of going for a bacteria, we can modify, introduce human gene into bacteria and produce the drug which we want. For example,
Which we want.
insulin. What is the function of insulin?
Produced by E. coli.
Insulin, see, it is a sugar, it reduces sugar level, blood sugar, reduces blood sugar level. Okay,
Yes, sir.
so in diabetes patient, the human gene is isolated, inserted into bacteria. Bacteria is unicellular organism, and bacteria now produce insulin. Insulin is administered to humans. It's a very, very important thing. Okay, that is genetic engineering. So that's another in medicine. We produce medicine, lot of medicine from bacteria, like insulin. Lot of medicine, not just antibiotics. Even insulin is also produced. Okay, and other factors like blood clotting factor. What is hemophilia? Hemophilia A, hemophilia B. Factor 8 is hemophilia. Factor 9. See, hemophilia means if any injury comes in that patient, no blood clotting will not stop. Okay, blood clotting will not stop. So for them, the clotting factor is produced from bacteria. They are injected. If clotting factor
So
8,
the higher
the
the
name,
number is, the more dangerous it is.
huh?
Like I don't understand what blood clotting factor 7 is. Like
Listen, see, if you get an injury, what goes out from your body?
what, sir?
Lose blood
Blood.
clear.
Blood.
Any injury, blood
Yes.
will be lost,
Yes, sir.
right? And all of a sudden, soon the blood leakage of blood stops. You can see clot there, clotting,
Like platelets, sir.
accumulation. Can you understand? Clotting of
Yes,
blood.
sir.
So, what causes clotting?
Platelets.
There are platelets and other chemicals. See, for the blood to clot, there are cascade of reaction. In higher classes, you study A should be converted into B, B should be converted into C. Like that, 12 reaction will occur. How many reaction chemical steps?
12.
12 steps occur.
12.
In that, for example, first step, factor 1. Second step, factor 2, factor 3, factor 4. This substance only carries out that process. In the 8th step, if in some patient, the 8th factor is absent. 8th factor is absent means what? Blood will not clot. That condition is called what? Hemophilia.
So that means
If the
for a
ninth
tiny
factor is,
blood, so that
huh?
means for a tiny wound to like stop the blood flow, all these 12 reactions happen. So
They have to take in the factor 8 medicine.
with no, like normal wounds and all, like for everyone, will all the 12 chemical reactions happen?
What?
For like you said, there's 12 reactions. Let's say for a blood to clot.
Uh,
Yeah, for us also, if we have blood like leakage or something, it'll take 12
hmm.
reactions to for the blood.
Everybody in every living organism, okay, even in animals, even in animal cattle, if they have blood, warm-blooded animals, same factor only. Okay.
Yes, sir.
Yeah.
Like
Sir, what
So,
we
happened if fish
Sir,
and all is bleeding, sir?
fish and all is what?
What will happen if fish is bleeding?
fish a warm-blooded animal.
in fish and all,
It's cold.
they
No,
have
fish is a cold-blooded animal.
Then
cold-blooded.
why it's
Listen,
same process
fishes
in
will have cold blood. They are not having water. Warm-blooded animals. We are
Like cold, no?
warm-blooded animals. They
Lay
have,
animals
see,
underwater are
the
all.
components are different. In fishes, the
Poolside.
components are different.
What do you
Sir,
mean, sir?
no,
There are,
whales
there
are
are,
warm-blooded. Whales are
Yeah, I
warm-blooded.
know that they're cold
see,
blooded. That
listen,
doesn't matter now.
animals you studied, right? That's what you didn't finish your animal kingdom content. Koi kilotoms, warm-blooded animals, cold-blooded animals. There, it is not going to be a big issue in fishes.
Yes,
Okay, they do not have heart, means they do not need circulation, means like us. Our system is different, their system is different. Okay,
Yes,
Yes,
sir.
the
sir.
composition different. So only humans, and especially this is human disorder, hemophilia. Okay,
So in fish,
Yes,
how
sir.
will it
human.
take place? So
Huh?
in fishes, how will it take place? Because they're cold-blooded animals, there'll be a different way for blood clotting, right? So what is the way?
In fishes, they follow different method. Okay, like
Yes.
always, they obtain the substances from, they're not eating like you, so they obtain the substances from water and then they circulate. The entire circulatory system is different. Okay, when we study about don't worry, this we study the circulatory system later. That time we will discuss. Okay, now we
Yes,
just
sir.
focus on this. Okay,
So isn't the
circulation is
circulatory
different.
system only for humans?
We will study the, at that time I will tell you the differences. No,
Oh,
please don't waste the time, you just listen.
yes,
Okay,
sir.
fine, don't worry, that we will see
Yes, sir.
now. Okay, vaccines. What are vaccines?
Antibiotics.
Vaccines,
Oh,
for
vaccine.
example,
Oh,
for corona and all, we used vaccines.
we
Okay,
like
so
in
Vaccine.
that,
vaccine and all, they have
yeah,
a
that
part of the
vaccines
virus.
are produced. Okay, for cholera, know the definitions,
So,
preparation of weakened bacteria, and
so,
then
what's the difference between antibiotic medicines and
then don't worry. Okay, see, listen, go through this chapter, come with your doubts, we will discuss in the next class. Okay,
So
fine,
vaccines are
because
just
lot more
like
things are there in this particular chapter. Maybe next week only we can finish. So please go through this chapter and come with all your doubts.
Okay,
yes,
sir. Thank you,
sir.
sir.
So,
Bye.
so wait.
No,
So like
we
for
will discuss in the
yes,
next
sir.
class, because we have lot of, we don't have time now, class time over. So definitely we will discuss all these things in the next class.
So I mean, we
Okay,
don't have a next class, so we can continue if you want.
Yes, sir.
okay, fine. Then you have to do self study only. Whenever we have the next class, we will study. Okay,
So small
otherwise
thing,
you will
Yes,
study.
sir.
Let
vaccines
me
are just substances which have a part of the virus which you want to destroy, right? Sir?
listen. There are different type of vaccines. Okay, some are weakened form, some are part. Okay,
Yes, sir.
based on that we classify vaccines in different way. Now we just understand weakened part. Okay,
Yes,
either
sir.
the
So
organism
like
or the
yes, sir.
products or whatever, or the modified project also.
So yes, sir. I heard my mom said when she got the vaccine,
That's what
don't
you
take
got
it.
to
Okay.
understand now about vaccines. Many things has to be said. Okay, so that we will
Yes.
discuss in the next class. If you want, I
Yes,
can
sir.
continue. Do you have any other class or not?
No, sir, we don't have one.
Okay.
We don't have any class, sir.
Okay, fine. Anyway, because I have another class, uh, it was not planned also to continue, but anyway, so don't worry, Pa. We will discuss
Yes,
all
sir.
lot more things are there in this particular area. Very simple thing cannot be just like that, cannot be said, because let me tell you how vaccines are prepared, what are the different type of vaccines, and how it is classified. All these things we have to discuss. Then only you can understand how vaccines are prepared and how it is classified. Okay, it
Yes,
takes time.
Yes, sir.
We just
yes,
stop
sir.
it here and then we will continue. Okay, it is good. Please go through once what we are going to see in next class. Then only it is easy for me to continue. Okay, and if possible,
sir.
let me ask them to increase the class as maybe if it is 1:30, 1.5 hours, will you be able to concentrate?
Sir,
Maybe instead
yes, sir.
of 5:30 to 6:30, can we prepone 5 to 6:30?
Sir, no, sir. Actually, usually we have two classes. Today, second class got cancelled.
Okay, okay, okay. But I have another class. That's all. Fine.
Okay, sir.
Okay, Pa. Then thank you. Take care. We'll
Thank
see
you,
you later.
sir.
Bye.
Bye,
Thank you,
sir.
sir. Bye,
Yes, sir.
sir.
Thank you, sir. Bye, sir.
Thank you.